In the last session, we reviewed why we treat the wastewater, how we characterize the wastewater, and a typical treatment train that is used to treat wastewater. Now, we're putting our focus on the biological processes that utilize microorganisms to convert the organic constituents to CO2, nitrogen gas, and biomass, which can be separated from the aqueous phase. Before diving into the fundamentals that find the commonality in bioreactors, I would like to introduce the diversity in biological processes in terms of reactor configuration, biochemical processes, and microbial communities. Based on the state of biomass in the reactor, there are two major types of bioreactors: suspended growth reactors and attached growth reactors. In suspended growth reactors, the biomass is in its planktonic state, suspended in the reactor. Conventional activated sludge, oxidation ditch, and sequential batch reactors are the typical suspended growth reactors. In contrast, the attached growth reactors, the biomass forms biofilm attaching on the surfaces. Packed tower, rotating disc reactor, fluidized bed bioreactor used to be the common attached growth reactors. But in recent years, granular sludge reactor, moving bed biofilm reactor, and membrane aerated bioreactor are getting more attention in research and industry due to their high treatment efficiency. In suspended growth reactor, because the suspended biomass is mixed with the bulk solution, there's no barrier between the substrate and the biomass. This results in efficient substrate uptake when well mixed, but mixing itself can be energy intensive. Also, to prevent washing out the biomass at short hydraulic retention times, the biomass needs to be retained and recycled with typically a clarifier, which is an extra structure that requires construction and maintenance. As for the attached growth reactors, because the biomass aggregates in biofilms and attach on surfaces, they're easily separated from the bulk solution and retained in reactor at high concentrations. Also, the biofilm acts as a barrier between the biomass and toxic substances, protecting the biomass from shock loadings of inhibitory compounds. But the biofilm also forms a barrier between the biomass and substrate. Substrates and oxygen need to diffuse through the biofilm to reach the biomass and to be utilized. Moreover, biomass sloughed or shedded from the biofilm needs to be removed or it will impact the effluent quality. Based on operating mode, the biological reactors, especially the suspended growth bioreactors, can be divided into three types: continuous stir tank reactor, CSTR, plug flow reactor, PFR, and batch reactors. CSTR is the most commonly used configuration. Biomass is completely mixed within the reactor while the reactor is continuously fed and vacated. Because of the complete mixing, the effluent quality equals the bulk solution quality. The influent is instantly diluted after being fed to the reactor. As a result, it can handle peak loads of organic matter and toxic compounds. But the low substrate concentration in the reactor also result in slow microbial growth and substrate consumption rate. This leads to a large reactor and high aeration cost. The low substrate concentration also favors microorganisms that are responsible for sludge bulking, like the filamentous bacteria. CSTR also offers limited operational flexibility. PFR is also fed and vacated continuously. Because there's little axial mixing, substrate concentration decreases along the length of the reactor as they are degraded. In reality, it looks more like this. This configuration results in a much higher average substrate concentration in the reactor compared to CSTR, hence higher biomass growth rate and substrate consumption rate. Therefore, a small reactor volume and less aeration is needed. PFR configuration also allows more flexible operations such as step feeding and zone aeration, which uses different aeration intensity at different zones based on the substrate concentration and biomass growth rate. But because there's no dilution like CSTR, PFR are more susceptible to peak loadings of substrate or toxic compounds. The oxygen demand at the inlet can be very high, although this can be mitigated by step feeding. But because of the flexible control options, PFR also requires more complex construction and higher maintenance demand. Batch reactors are like temporal PFR, or PFR over time. After filling the reactor with substrate and biomass, there's no more feeding or wasting. Substrate concentration decreases over time as it is consumed by the biomass. Batch reactors are frequently applied in the form of a sequential batch reactor. In sequential batch reactors, when substrate concentration is reduced to the desired level, the biomass is settled so that the clean supernatant can be decanted. Then the reactor is ready to be filled with next batch of substrate. Batch reactors allow single tank reaction and operation. There's no need for a second clarifier. It also allows flexible operation such as overtime aeration intensity adjustment and substrate step feeding. Like PFR, batch reactors do not dilute the feed, therefore is also susceptible to peak loadings of toxic compounds. Also, initial decant may contain solids that does not settle well and may need to be recycled back to the reactor. This can reduce the actual capacity of the reactor. A more flexible and sophisticated control system also means higher operational cost associated with higher maintenance demand. So we have talked about the features, the pros and cons of the CSTR, PFR, and batch reactors qualitatively, and we'll discuss their mathematical models quantitatively in a later class. There are some other configurations of attached growth reactors. I would like to introduce a few of them here. Attached growth reactor may have configurations that are different from the conventional CSTR, PFR, and batch reactors. Pack tower is an example. Wastewater is sprayed on the packing material and is degraded as it is trickling down the biofilm grown on the packing material. Even though it is mechanically simple and very energy efficient, it is very hard to design and model as a hydraulic condition, and the formation of biomass in the packing material are very difficult to determine. In fluidized bed bioreactor or upflow sludge blanket or granular sludge reactor, a granular type biofilm is formed around a carrier material, which can be sand or activated carbon. An upflow and air suspends the sludge granules in the reactor, degrading the substrates. The sludge granules result in a very high biomass concentration in the reactor, hence high substrate degradation rate and high reaction efficiency. But it requires precise hydraulic control or risks losing sludge granule and carrier material. A designed carrier material can also be used instead of sludge granule to provide a high specific surface area to enhance the contact between biofilm and substrate. This is the moving bed bioreactor. Of course, I did not cover all the configuration there are, but you can find information about a lot of them in Table 1.2 and throughout the DDLF book. You can also think about their pros and cons and their ideal application based on their design and things you will learn in this course. In addition to reactor configuration, a variety of biochemical processes occur in the bioreactors treating wastewater. One of the major reactions in conventional wastewater treatment is the aerobic degradation of hydrocarbon. We will later look into the energy flow of this process in more detail. Biological nutrient removal includes biological nitrogen removal and biological phosphorus removal. In biological nitrogen removal, the organic nitrogen and ammonia are converted eventually to nitrogen gas, while in biological phosphorus removal, phosphorus is uptaken by phosphate accumulating organisms, which can be separated and removed from the aqueous phase. We will also cover them in more details later in the semester. Anaerobic processes play an important role in recovering resources and energy from wastewater. It converts the organic carbon in volatile fatty acids, hydrogen gas, and eventually to methane gas. Medium-chain fatty acids can be used to produce biodiesel and biokerosene. Volatile fatty acids are good carbon sources for denitrification. Hydrogen gas is a good energy carrier and reducing agent. And eventually, methane gas can be used to produce heat and electricity. We will also talk about the details of the anaerobic process later. All these biochemical processes are performed by diverse microbial communities. Using advanced next-gen deep sequencing, thousands of the species were discovered in a bioreactor treating wastewater, many of which are previously uncultured and unknown. We are starting to slowly understand their functions and roles in the bioreactor ecosystem, and are placing them on the tree of life. In the domain of bacteria, we know there are heterotrophs that breaks down organic carbon aerobically and anaerobically, like Acinetobacter and Clostridium. There are also autotrophs like ammonia oxidizing bacteria and nitrite oxidizing bacteria. In the archaea domain, methanogens converts acetic acid or hydrogen and CO2 to methane gas. In eukarya, fungi can hydrolyze and degrade complex hydrocarbons, while protozoa can graze colloidal particles and disperse bacteria. Viruses are not on the tree of life, and little is known about their roles. They may be involved in the lysis of cells and horizontal gene transfer. Next, we are going to unify all these diversity in biological processes using energetics, stoichiometry, and kinetics.